So in terms of our lecture format, I'm just going to talk briefly about learning objectives for this lecture, and then a little bit more detail just on the study guide, just in case anyone wanted some further tips. Again, just kind of generally how to study physiology as a subject. And then I'll talk about, then I'll dive straight in, sorry, to cellular physio. So these are the topics which have been broken down, which we've broken down cellular physiology into. So structure and function of cell membrane, organelles, this is a recap from biology, in addition to transport of substances across the membrane, again, recap from biology and also histology, compartmentalisation of body fluids and the differences between the fluids in the different compartments, very important for physiology. Then I'll talk a bit about ion channels, and then we'll go into some of the electrophysiology, so resting membrane potentials, action potentials, and then a couple of the definitions, excitability, refractoriness. And then I will conclude this lecture by talking a little bit about musculoskeletal system physiology. So I'll talk about the NMJ, I'll talk about excitation-contraction coupling, and then just molecular mechanisms behind muscle contraction. So all of these topics, I would say these are kind of general physiology topics. These should be building or should really build on your knowledge, pre-existing knowledge from other subjects like biology, biophysics, histology as well. So this lecture is not extensive. The idea we had was to provide a rapid review, as the name of the lecture series suggests. So it's not extensive or exhaustive. It's just here to provide you with the kind of core information for each part. So in terms of the learning objectives for this lecture, so I want to just, as this is our first lecture, I want to give you an overview of how to kind of just study physiology, but also before I dive into the topics, and then a few things I would really recommend. First of all, if you are studying physiology for your exam this summer, it's a very, very good idea to make a weekly timetable or a daily timetable. Most importantly, make it realistic and include recap time. So, just be prepared to spend twice as long or even 3 times as long on a topic that you plan to study for a week. Perhaps may take 3 weeks or 2 weeks. It just really depends, but you won't know until you start. So most important thing is starting, and then maintain discipline. Maintain discipline. So you're basically, you have a very strong start now, and then nearer the time of the exam, you're in a much more confident, comfortable position to be recapping the material, because as you all know, physiology is a huge subject, and it takes a lot of recap time, a lot of revision time. Point number 2 is quite important, and it will vary from faculty to faculty, but I would just really recommend you make sure you have all the materials you need, all the materials recommended by your lecturers and senior medical students at your faculty, just so you ensure you're getting the right information, the correct information, and you're learning to the correct level of depth needed for your particular university. So as a whole, I said I would talk to you about kind of how to study, sort of the building blocks. So just I glossed over it in the last slide, but first points would be, first of all, gather your materials, talk to your upper year medical students. These guys are past physiology. You may even have students who are also assisting the department, either in teaching or in research. So they're in a really good position because they, talk to the professors and stuff. So they have, pretty much the best advice to give regarding the pitfalls, common pitfalls, do's and don'ts, key areas to focus on. Those kind of bits of advice, quite important. Discover what methods work best for you. Yeah, this one's a really important note, because not every study technique is suited to every individual. It's quite unique, and therefore study patterns and study revision should be tailored to the student. So I would recommend try a few options, try a few different techniques: individual study, group study. Obviously, at the moment, it's not, you can't go to the library, but it could be group study on using a video conferencing software, for example, speaking out loud, writing notes, just try all these different techniques and see what works best for you. Don't be dissuaded by bad advice. Yeah, that's an important one as well. You might hear, don't study this book, or for example, this certain teacher likes this or that. I think don't be dissuaded by any bad advice. Just go into it with a clear head and with the intentions of studying everything from the topic, leaving no subsection left. Study primarily from the material which is recommended by your faculty. That's obviously important. A good textbook is always recommended as a reference of physiology. So a few off the top of my head I can remember, well, the one I used was Borons. Other recommended textbooks would be Guyton's or Ganong's, and there's a couple of others as well, so BRS Review or Constanza. But it's generally a good advice to have a reference book on hand. And any additional literature is also useful for refining your knowledge. If you read something in the textbook or you read something in your lectures you don't fully understand, there's a bunch of there's so many YouTube videos or student-made documents from your faculty which can help, which can point you in the right direction. I mentioned this again: recap material continuously, and obviously decide what type of study works best for you. And then finally, test yourself. So repeated testing, active recalling what you've learnt, and make sure it's done, it's done after a period of a few days or a week, because I guarantee you'll learn something 1 week, and then by the next week you'll be moving on to the next topic, and it's very easy to forget the content because you're covering so much material. It's always important to kind of just take stock on what you've learned every kind of so often. You decide when that is. Stop and think about what was covered in the previous week or what was covered 2 weeks ago, for example, so continuously recapping and testing yourself. And that's the best way to kind of integrate all this content you're learning into your long-term memory. So yeah, create a checklist is what I mentioned before, of weakest topics, and kind of rank them. So if you're someone who has studied physiology before and you're repeating the subject, or you've studied it last semester and you kind of want to know where to start this semester, I'd recommend breaking down all the topics. So for example, general physiology, cardiovascular, renal, GIT, respiratory, endocrine, etc., and just rank them in terms of your weakest topic first, and start with that one. Finally, if you can teach a topic in physiology, you know it well. I really believe this for all medical subjects, but I think physiology most of all. Physiology and biochemistry are preclinical subjects which require in-depth understanding. It's not just about memorization. You really do need to understand the concepts. And the best way is to teach or speak out aloud. So either with yourself or with a partner or with a friend or a group of friends, I would really recommend this if you don't already practice this, just to try out. Okay, so let's dive in. So the first topic from cell physiology is concerned about the structure and functions of cell membranes and organelles. So I've broken this down into 2 slides. As I said before, this lecture is a rapid review series. We're not going to be extensively going through every last bit of detail, but I will try to highlight the most important points. If for any reason you're watching this and you are confused about something I've written in the slides, or you're confused about something I've said, or you want a bit more clarification, obviously, look at the resources that you have, textbook materials, online resources, but I'm more than happy for you to contact me, and we can message and talk about it, or we can have a chat about it. I don't mind. I'm really open to that if you need it. Okay, so let's dig in. So structure and function of cell membranes and organelles. So membranes themselves, first of all, and then organelles. So membranes are composed of 2 main structures. We have our lipids and we have our protein components. So lipids, namely phospholipids, have a glycerol backbone, which is the hydrophilic or water-soluble head, and then there are 2 fatty acid tails, which are hydrophobic, so water insoluble. The hydrophobic tails will face each other and form this bilayer. Lipid soluble substances, so for example steroid hormones, your catecholamines, for example vitamin D or vitamin D analogs, these substances, because they are hydrophilic, can dissolve through the lipid membrane. In addition, we also have certain molecules like oxygen, carbon dioxide, which are small molecules which can also cross the cell membranes themselves quite easily. In contrast, water-soluble substances such as sodium ions, chloride ions, macromolecules such as glucose and water as well, cannot freely dissolve in the lipid membrane, but they have specific channels or specific proteins which allow or facilitate their transport, which we'll discuss later. So then the protein component. So protein component, we have integral proteins and peripheral proteins, one way of dividing them. Integral proteins are anchored to and embedded within the membrane through hydrophobic interactions, and they may span the length of the cell membrane. Typical examples of integral protein would be ion channels, transport proteins, receptors, guanosine 5-trisphosphate binding proteins, or GTP binding proteins, or just simply known as G proteins. In contrast, peripheral proteins are not embedded in the membrane, so they just very loosely or covalently bound to the membrane components, and they're bound by electrostatic interactions. And finally, we have intercellular connections. So these are connections between adjacent cells. So two main types we have are, and this is again recap from histology, so tight junctions or zonular occludens in Latin. These are the attachments between cells, often epithelial to epithelial interactions, and they may be an intercellular pathway for solutes to travel. And we have different subtypes as well, so tight versus leaky as well. And depending on the localization, depending on the function as well of that specific tissue type, we can see different types of tight junctions. So for example, in the renal tubules, we find these tight junctions. In gap junctions, so gap junctions are slightly different. These are larger gaps, larger junctions between adjacent cells to permit, again, intercellular communication. Typical example here is between myocardial cells, where we have current conducting through the working myocardium and the conductive myocardium, sorry. So we need to have gap junctions in abundance to allow for the rapid flow of current through the myocardial cells to allow for excitation and then contraction. So on the next slide, I've got a bit more information, and I've got a nice scheme here which summarizes what I mentioned before. This is a bit about the function of the cell membrane. So just very generally, one of the main functions is compartmentalisation, which we will discuss in a moment. The membranes serve for kind of holding the contents, the organelles, and also the liquid material, all the ions and the macromolecules in their own intracellular environment. So they form these compartments within the body. And this is very important for a few reasons, which I will discuss in a later slide, why we have these compartments. Another one is permeability. Permeability, so for the cell membrane, we say is permeable for specific ions at different times, which allows the passage of various ions and substances through. Fluidity, endocytosis, pinocytosis, receptor-mediated endocytosis. Obviously, semi-permeable membrane is essential for maintaining the resting membrane potential. We will talk about this too in a moment. And this is all facilitated through the proteins which are embedded in the membrane. So maintenance of the resting membrane potential is maintained through, is kept through the very, very important sodium-potassium ATPase, which pumps 3 sodium ions out of the cell for every 2 potassium ions into the cell. And also permeating lipophilic and lipophobic particles as well. In terms of the structure and function of organelles, I've just obviously listed the main ones here. Again, this is a review over biology, so I'm not going to talk about this in detail. So nucleus, as you know, it's important to obviously know the function, structure and function for all of these things. So structure is, I think, quite self-explanatory in terms of function: nucleus, cellular machinery, cellular brain, mitochondria, powerhouse, ATP production, oxidative phosphorylation, aerobic respiration. Rough ER, smooth ER, very similar. Rough ER is studied with the ribosomes, smooth ER is not. Both very important for processing proteins. So we have our translation, transcription, and then we have our post-translational modifications, which take place in the endoplasmic reticulum. Golgi apparatus, again, is very important for lipid processes, lipid processing, and also packaging of any components which have been made in the cell. Lysosomes containing various degradative enzymes, and ribosomes, like I mentioned before, very important for the protein synthesis, proteosynthesis. Okay, so we'll move on now to the transport question. So transport of substances through the membrane. So there are various different methods that substances can be transported through our cell membrane. So I've broken them down here for you. So first of all, let's talk about simple diffusion. So simple diffusion occurs down a concentration gradient, and it's a passive process, which means it does not require any metabolic energy. We have a high concentration in one area, and we have a lower concentration in another area, and it's just passive. It's just passive movement. High density of molecule will naturally spread and dissipate to areas with lower concentration or lower density. And it can be modeled with this formula here. So J is equal to PA multiplied by the concentration of A minus the concentration of B. So J is the net rate of diffusion, or we can say flow. So this is in millimoles per second. So for those of you who are attentive, you can see that this is a concentration per unit time, and is equal to the permeability multiplied by the surface area for diffusion. This is multiplied by the difference in the concentration. So we have, obviously, like I said before, concentration in part A, concentration in part B. And this is how we can calculate diffusion. We end up with a rate, so we can determine how fast a specific solute will diffuse from one area to another area. And facilitated diffusion is similar in the sense that it's also traveled down, the solute travels down its concentration gradient, or its electrochemical gradient. So it's also a passive process. However, it requires a protein. In simple diffusion, no protein is required for transport. Whereas in facilitated diffusion, as the name suggests, we are facilitating the diffusion process. So we have a carrier protein. For example, glucose I've just written glucose transport in muscle cells, but there are a few examples, and it's important for these general physiology questions to have a few examples on hand for all of these different types of channels and types of transport. So in contrast now, so we've talked about simple diffusion and facilitated diffusion. Now let's talk about active transport. Active transport can be broken down into primary and secondary. So primary is the one which you're probably most familiar with. This is against an electrochemical gradient or a concentration gradient. This means that we have the movement of a low concentration solute to an area where there is higher concentration. And this requires metabolic energy. So in the human body, we get this energy from ATP, adenosine triphosphate, and this is active transport. A typical example would be the sodium-potassium ATPase. We have many other examples in the body, but this is the most notable one. This is the most important one. It's found in every single cell. And as I said before, it's one of its responsibilities, one of its roles is to maintain the resting membrane potential. Secondary active transport, so in contrast to primary, it's the transport of two or more solutes. And what happens is that the movement of these solutes are coupled. So we have one solute transported down its concentration gradient. So it's, for example, sodium traveling from high concentration to low concentration through a channel. And what happens is this movement down the concentration gradient actually provides energy, and it provides enough energy for the transport of a completely different solute to travel against its concentration gradient. And a typical example is what's called NCX, or sodium calcium exchanger. Another example, sodium glucose cotransport. And you'll find in renal physiology, we have a lot of cotransporters, and these are typical examples of secondary active transport, when a solute moves against its concentration gradient simply because enough energy is produced by another solute moving down its concentration gradient. So secondary active transport can be either antiport, which means something moving from let's say a solute moving from A to B, and then another solute moving against its concentration gradient from B to A, so antiport. Secondary active transport can also be symport, where we have movement in the same direction. And these are examples of co-transport. So another couple of examples, osmosis and filtration. So osmosis, this is how water molecules obviously move through a semi-permeable membrane. This requires no energy. It's a completely passive process. And eventually over time, water will equalize in the concentration of these two solutions. Filtration is the movement of a solvent as a result of osmotic and hydrostatic pressure. So we need to talk more about, or rather a point for you guys to look into is when you study filtration, look up Starling forces, look up your resorptive and interstitial fluid definitions, be really familiar with hydrostatic pressures, oncotic pressures. Okay, so let's move on to the next question. So the next topic is on the compartmentalization of body fluids. So I mentioned before, this is one of the functions of our cell membranes. Probably our main function of cell membranes is to keep everything where it needs to be. And there are a few reasons we do this. So the next topic I'll talk about after this is the differences between intra and extracellular fluids. But let's take a step back for a second. Let's look at all of the compartments we have. And I really like this scheme because it breaks things down quite nicely for us. And values, when it comes to values, such as percentages or units, just please bear in mind that the numbers will vary from source to source. I've tried to quote the figures from, I've tried to take figures from as many sources as I can and try to use the most frequently used figures, but they will vary from textbook to textbook. So this scheme shows us quite nicely that we have total body weight here in blue or bluish green color. And we can divide up the total body weight quite logically into solids and liquids, or solids and fluids. And this is very important to remember, 60% of the total body weight, or TBW, is liquid, is fluid. The remaining 40% is something which we're not concerned with for this question, but it's solid material. That's quite a lot of fluid in the body. It's very, very important to understand that. So from this fluid portion, we can divide up very interesting, I think it's quite obvious, obviously, into intracellular and extracellular fluid. 2/3 of that is intracellular, inside the cells. 1/3 is extracellular. From that 1/3, 3/4 is interstitial fluid between the actual cells themselves in the tissue, and 25% of the extracellular fluid is intravascular, intravasal, so plasma, essentially. This is basically a good overview of where the liquids are stored, and it's very important to remember these numbers and these ratios. Some other definitions, so transcellular fluid, you may hear about. This is, again, extracellular fluid. This could be, I've put a few examples here. It's not an exhaustive list. So GIT secretions, cerebrospinal fluid, peritoneal fluid, pleural fluid, ocular fluid, synovial fluid in the joint cavities, in the diathroses. These are all transcellular fluids. Third space, this is pathological. Third spaces, these are volumes in the body which can be occupied by liquids, but they shouldn't be occupied by large quantities of fluid. And then changes of distribution of the values in this table will lead to pathologies. So edema, dehydration, vomiting, occupation of the third space. So just important to know the compartments and what we find or what we expect to find in each compartment. So now let's talk about the differences between intra and extracellular fluids. So I mentioned before a few times, the membrane itself creates the barrier. Yeah, the cell membrane is what's creating the division between intra and extracellular fluid. So distribution of ions and proteins, intracellularly we have a negative potential, extracellularly is a positive potential, and our resting membrane potential is the difference between the two, which is the membrane potential of the actual phospholipid bilayer, which is maintained by the transfer and constant moving in and out, the constant movement, of ions. So yeah, as I mentioned before, constant movement of ions, and this is what's going to cause our excitation, which I'll talk about in a couple of slides. So proteins will differ intra and extracellularly. We have plasma proteins such as albumin, thyroglobulin, we have corticosteroid binding globulin. We've got many other proteins in the body in different compartments. I think the most important thing to understand from the differences between intra and extracellular fluids are the differences between the ions themselves, or the electrolytes. So you can see quite nicely in this table, we have extracellular fluid, ECF, cations and anions. So if we compare ECF cations with ICF cations, we can see in the extracellular fluid, we have an abundance of sodium. And we have a very low concentration of potassium ions. In contrast, intracellular cations, the most abundant is potassium, and sodium is at a very low concentration. This is essential because It's the influx of sodium which is going to cause our cell membrane to depolarize, and we have the highest concentration of potassium inside the cells. Cellular damage can lead to, or cellular rupture can lead to hyperkalemia because of this reason. This is how it can be explained. Potassium is stored in high concentrations in the cell. In terms of the anions inside the cells, so intracellularly, we have a lot of phosphates, we have a lot of bicarbonates, proteins, but the most important thing here is you can see phosphates. In contrast, the anions we find in the extracellular space will be chlorides and bicarbonates. So main difference there, chloride versus phosphates. It's not important to know the numbers by heart. It can be helpful to learn sodium, potassium concentrations, intra and extracellularly, but it's not important to know these numbers all by heart. Okay, so a bit more detail now about how these ions are transported. So ion channels themselves. So these, as I mentioned at the very beginning, these ion channels are an example of integral membrane proteins, which means that they span through the entire depth of the phospholipid bilayer. And there are a few different categories and types. So ligand-gated channels. Ligand-gated channels are or metabotropic are acetylcholine, typical example of a transmitter which is conveyed, which binds to a channel to open it. GABA and glycine, again, other key transmitter molecules. And what they will do is they will bind, as you can see in the scheme at the top. These substances will bind to the extracellular domain of the ion channel, and they will cause the change in configuration of the ion channel, and then the ion channel will open or it will close to allow the passage of ions through the channel. So ions, notably calcium, sodium, potassium, or non-specific cations, for example. So another type is voltage-gated, which means that it's not a ligand binding which causes or influences the opening or closing of the channel, but it's the change in membrane potential. So typical examples: sodium channels, calcium channels, potassium channels. the letter I meaning current, and H is denoted as for funny current. So this is statistics for creating what's called a prepotential in the sinoatrial node. So stretch-gated channels, in contrast to the two types of channels we mentioned before, stretch-gated will open as a result of mechanical stress. So typical examples: baroreceptors, stereocilia in the inner ear, and then we also have some other types of leaky channels as well. A bit of detail which I would recommend looking into. So we have these channels, as I mentioned. We have various ways of activating them, deactivating them, but they also have, and this is I guess a bit more. This is where it gets a bit more complicated. They also have activity. They have active and inactive gates, and substances can bind to an active gate or an inactive gate. It's just something to look into. This is how they can be regulated. Okay, so let's jump to the next slide, because the next slide will take a bit of time to go through. So action potential. So this is the graph, which I would really recommend you learn off by heart. Should be, ingrained. It should be your bread and butter to know how to draw an action potential, a basic action potential in neural tissue. Also learn how to draw an action potential for conductive myocardium. So typically you can use the SAN, and also learn the graph for working myocardium, so ventricular myocyte, for example. I think those three graphs are pretty essential to know. Okay, so an action potential, first of all, what is it? We need an action potential to elicit an electrical response and to conduct electric energy for a specific purpose. It could be conductance of a neuronal impulse. It could be an action potential which will go on to elicit muscular response. Those are the two main types. So how does it work? How is it broken down? Well, we have different phases, and it's very important to know the phases, and it's important to match up the phases to the graphs. So depolarization, first of all, makes the membrane potential less negative. So the cell interior becomes less negative. Hyperpolarization makes the membrane potential more negative. So the cell interior becomes more negative. An inward current is the flow of positive charge into the cell. An inward current will depolarize the cell. An outward current, to the opposite, is the flow of positive charge out of the cell. So outward current hyperpolarizes the membrane potential. Okay, so like I said before, an action potential, an AP, is a property of excitable tissue. So nerve cells and muscle cells we talk about. So we have rapid, very, very fast depolarizing phase or upstroke, which is then followed by repolarization of the membrane potential back to its resting membrane potential, RMP. Action potentials have a set configuration, as you can see in this graph, and they exhibit a all or nothing response. So I'll discuss what this means in a second. So as you can see from this graph, the resting membrane potential is set at minus 70 millivolts. This is our basal level. This means that at this voltage, the cell is able to excite. It's ready to excite. You can see, the one reason I like this graph is because you can see a subthreshold stimulus. So this is basically opening of some sodium channels, but not many, but just a few, let's say. It's difficult to quantify, but the resting membrane potential increases slightly to, on this graph, I would say to approximately minus 60, minus 65, somewhere there. But the intensity is not enough to elicit a full action potential. And this means that the threshold hasn't been reached. The threshold is this dotted line you can see, which is marked at about minus 55 millivolts. So this is what I mean by all or nothing response. We have to have opening of a huge number of sodium channels, an influx of a very high concentration of sodium into the cells in order to cross or to reach this threshold potential, threshold level at minus 55 millivolts, after which the depolarization will occur and we have our action potential, we have conductance. Anything under this is called a subthreshold stimulus, which means there is no action potential. I just want to make that clear, that this graph, this is not part of the action potential. This is just illustrating that a subthreshold stimulus will not cause an action potential. This is our action potential. Okay, so in terms of the phases, so like I said before, inward current, sodium channels open, depolarization of the membrane, exceeding the threshold potential, and we reach our peak at about minus plus 20, plus 30 millivolts. After which point the sodium channels will close and repolarization begins. At this point, sodium channels will all close, potassium channels will open, and this means that potassium will increase, potassium concentration will increase, and sodium, which was in the cells, will need to be removed from the cells. And this is done very efficiently by our sodium-potassium ATPases. So bringing 3 sodium ions out of the cell and replacing them with potassium ions, 2 potassium ions. After the depolarization has occurred, we have what's called our refractory period. So there's another slide coming up. I think it's the next one, which will talk about the types of refractory periods. So let's talk about that now. Okay, so excitability, first of all, just a very general definition. It's the ability of tissues to be excited by a stimulus. So they could be neurons, muscles, or sensory cells. I mentioned before what a subthreshold potential is. And you can see a bit of detail there. I would really recommend looking up these terms, EPSP, IPSP, EPP, and a prepotential. In terms of stages of the channels, this is a bit more detail again about the activated and inactivated forms of the channels. But the most important thing here is the refractory periods. So I'm actually going to just go back one slide. And I want to just show you on here. So after depolarization has occurred, we're over here now, then we have repolarization. Now, different texts will differ in terms of when they say that the refractory period begins. Some texts will say it starts somewhere here. Some texts will say it starts during the repolarization phase when the sodium channels are closed, potassium channels are open. So it's important to know that it happens after depolarization. Okay, so somewhere along this downward slope of the curve. And what this means is that, sorry, I should have mentioned actually, the absolute refractory period is what happens first. Absolute, because it means that during the absolute refractory period, we cannot have another action potential. The cell is polarized. Cell membrane potential, the cell membrane needs to return back to a normal level. That's why it's called absolute. But we do have a small period which occurs after the absolute refractory period, which is called the relative refractory period. And during this time, an action potential can occur if the stimulus is strong enough. So now let's look at this again. And this kind of just summarizes what I said. So when the voltage-gated channels are inactive, cannot be excited again by any intensity of stimuli. So this is what's called absolute. However, during the middle of repolarization and the later part of repolarization, if the stimulus is strong enough, like I said, then we can have another action potential. This is what's called the relative refractory period. So refractoriness is defined as the insensitivity to further stimulation. Okay, so it's just really important to understand when those two phases occur and why they're there. Basically, they're there to protect the cell. If we didn't have this system in place or this parameter, then the cells would be able to continuously excite, self-excite, and obviously this is not efficient. This would obviously lead to various pathologies, and we need this time lapsed after excitation to restore the ionic disbalance, because we have a high concentration of sodium ions intracellularly. They need to be removed from the cell. So we need a bit of time to remove the sodium ions and to restore the balance of ions. Okay, so now I have a couple of slides on musculoskeletal physiology. So kind of just following on from what we've just talked about with the electrophysiology. So I just want to talk quite briefly about neuromuscular junctions. So a neuromuscular junction is the interface between the nervous system and the muscles. Now you'll know this from histology and also from biochemistry. So I won't go into too much detail. These slides are a bit wordy, but I just wanted to make them wordy just so that you can see the sequence of events that occur. Because with these questions, it's all about a sequence of events. You know, something happens to event A leads to event B, which leads to event C. So it's just nice to follow the events like I've kind of just written, basically. So for depolarization at the NMJ, the neuromuscular junction, our ligand binding is acetylcholine. Acetylcholine binds to its nicotinic NM-type ligand-gated ion channel. So we talked about this before. ACH binds to the ion channel. This opens the channel. This is what we need because this is our ligand-gated channel. So binding causes opening, and now we have a sodium influx because the channel is now open, and we have potassium efflux. So sodium is now entering into the cells at the end plate. This is the end part of the neuron. This causes what's called an end plate potential, where we have our voltage-gated sodium channels will open, and we have creation or generation of an action potential along the membrane. And this spreads the positive charge through the membrane inside the cell. In the cell itself, we have activation of a ryanodine receptor. The ryanodine receptor causes the release of calcium into the cytosol from the sarcoplasmic reticulum. Now, this is a very complex system, which is coupled with what's called a transverse tubule or a T-tubule. So it's a good idea to revise the biochemistry behind this before trying to study this question. Repolarization occurs similarly to in neurons, where we have inactivation of sodium. We have activation of potassium channels, so we have efflux of potassium which leads to the increase of electronegativity in the cell, quite logically, and then essentially degradation of acetylcholine. We need to recycle the acetylcholine, so this is broken down by acetylcholinesterase, and is reused, essentially repurposed, repackaged into vesicles. So in terms of muscle contraction, this is something which I will discuss in a bit more detail in the next couple of slides. So I'm not going to talk about it right now. But essentially, quick recap: neuromuscular junction. It's a specific type of a synapse cell-to-cell interaction, but it's not between neuron and neuron. It's between neuron and a muscle cell. Could be smooth muscle, so leiomyocyte, or it could be skeletal muscle, so rhabdomyocyte. So in terms of excitation-contraction coupling, this essentially talks about how do we pair these two processes together. We have excitation through the nerves, and then we have contraction in the muscles. So, you know, what connects them? So essentially, it's a recap over the previous question discussing the neuromuscular junction, which you can see. It's explaining how calcium is released from the sarcoplasmic reticulum into the cells. And then it's talking about how the calcium itself, you need to mention how the calcium itself leads to contraction. So this is about the molecular mechanism behind muscle contraction. So the next slide, the last slide, we'll talk about that. So I won't mention that too much, but as you can see from this slide, there's a lot of recap between what I've already talked about. In terms of actual muscular contraction, it's important to mention that a contraction occurs after calcium binds to troponin C, which is released from tropomyosin and causes a power stroke. So like I said before, I'll discuss this in a second. And electromechanical latency, or EML, is the time delay, or the time which is between the initial stimulus of the action potential all the way until the onset of contraction. And there's a delay there for the movement of ions, and is caused by the time taken for calcium essentially to leave its sarcoplasmic reticulum through its channels, and then initiate contraction through the power stroke. So let's talk about the molecular mechanism now. So again, recap of biology and histology. So sarcomere units, very, very important to know this scheme. Just keep practicing. Just draw out a simple scheme like this. Okay, so what are the main things to understand from this? Well, a sarcomere extends from two lines, or discs, we can say. So these are our Z lines or Z discs. Muscle tissue is made up of, this is not, obviously I'm not going to talk about detail behind what muscle tissue is made up of, but just very simply it's made up of myosin and actin filaments with other associated proteins as well. On our sarcomere scheme, you can see that there is an A band at the bottom of the scheme. So A band is the entire length of myosin. In the middle of the A band, or the sarcomere, we have the M line. I band is in between one myosin chain, or between the myosin filaments, sorry. The Z line is the middle of the I band, as you can see. And these are just pretty abstract, but important to memorize. And finally, the H zone or H band is the part where we only have myosin present, because as you can see from the scheme, every other zone contains overlapping of myosin and actin. So the contraction itself, so before contraction, it's important to remember that ATP is bound. Yeah, it's bound to the myosin head, but then gets hydrolyzed to adenosine diphosphate and inorganic phosphate. Adenine diphosphate and inorganic phosphate. This itself, the process of this cleavage will release energy, causing the head to stroke backwards. So the reaction itself causes the movement of the head, like a spring. And as it does this, it reveals a new site for binding, basically. And when the ADP is released from the molecule in its bound form, the myosin will return back to its original position. And essentially this continues to happen, this through the continuous cycling of ATP, we have continual power strokes, continual muscular contraction, as the actin and myosin slide over each other as they contract and then relax, contract, relax. And it's all due to the expenditure and the usage of ATP, our energy currency. So it's hydrolysis of ATP, which is powering this process. So I'd recommend really looking at the scheme, understanding the different components. And again, there's a lot of overview here with biochemistry, so please review biochemistry, muscular biochemistry, because I think it will give you a good foundation here. Okay, so that's my high yield rapid review lecture for cell physiology and a bit of musculoskeletal physiology. If you have any questions at all, like I said before, please drop me a message. If you've got anything you'd like me to clarify. If not, I really hope you guys study well. I wish you all the best of luck for physiology, and yeah, I'll be giving a respiratory physiology lecture in the coming weeks. So hopefully you enjoy that one too. If I could please ask for you to fill in this Google form. It will take literally 30 seconds to a minute. It won't take long at all. It's completely anonymous, and it's really useful for us as an organization, but also me as a lecturer as well, to understand what went well, what I did well, what I could have done better, and for UIMS as well to assess the quality of teaching and also the syllabus as well. So if you guys want anything changed, this is the best chance to make your voice known. So that's all from me, and yeah, I hope you guys have a nice day. So yeah, take care.